My name is Katina Langley, Medical Science Liaison with Reckitt Medical and Scientific Affairs. Thank you for joining us today for our special segment on advances in cow milk protein allergy and the role of the microbiome. Please welcome our special guest, Dr. Fayed Ghishan. He is the chair of the Department of Pediatrics at the University of Arizona, the Horace W. Steele Endowed Chair in Pediatric Research. Dr. Ghishan is a board-certified pediatric gastroenterologist, and he is internationally recognized as an expert in gastroenterology and related basic and clinical research. His laboratory has been in the forefront of the characterization of the microbiome in relation to nutrition or nutrient gene expression and autoimmune disorders such as inflammatory bowel disease. He has authored more than 250 original publications. He's received 2 prestigious merit awards from the National Institutes of Health. Currently, he is at the University of Arizona as a professor and head of the Department of Pediatrics and the director of Steele Children's Research Center. Dr. Ghishan was previously the director of the Division of Pediatric Gastroenterology and vice chair of research at Vanderbilt University. Please welcome Dr. Ghishan, and thank you for being here.
Katina, thank you so much for this kind introduction. I'm so delighted to be with you here in your Salesforce meeting, and I'm so glad that I have met many of you in the last couple of years, giving lectures around the country on this role of the microbiome and cow's milk allergy. Thank you again for being here.
So, Dr. Ghishan, can you tell us a little bit about what is the microbiome? This term is used a lot. Can you explain that to us?
That's a very good question. The microbiome is the collection of genes encoded by the bacteria in our gastrointestinal tract. Microbiota is a collection of the bacteria in our GI tract. So the truth is, we are, as a human, we are a super organism. That is, we have another organism living in our GI tract. The number of bacteria in our GI tract is 100 trillion bacteria is present in our GI tract. The number of genes encoded by the bacteria is 3.3 million, and in fact, our whole genome is only 23,000. So literally speaking, 90% of our DNA is bacterial DNA, and 10% of our DNA is a human DNA. We, as a human, need to live in harmony with the bacteria in our GI tract. And that's the reason why the microbiome plays a major role in our health and disease.
So can you explain to us a little bit about how the microbiome impacts infant health?
So the microbiome is so essential in our health in regard to signal coming from the microbiome to allow our immune system to develop in the gastrointestinal tract. Let me explain to you this briefly. 70% of our immune cells are present in the GI tract. So those immune cells, they get their signal from the microbiome. So having a good microbiome allow you to have a very good immune system, as well as your integrity of the epithelial lining of the gut is very relevant with the microbiome being in a good shape.
Okay, so there are several terms today that are used that refers to bacteria. So prebiotic, probiotic, postbiotic. Could you please explain to us the differences between all of those?
Thank you. So probiotics, pro is for, biotic life, for life. Probiotics, by definition, is a bacteria which produces benefit to the host when it's given in adequate amount. The prebiotics are the substrate, or simply speaking, it's the food for the probiotics to survive on. So remember, you cannot have a probiotic surviving in your GI tract without prebiotics. So prebiotic and probiotics are both component of our gut microbiome. Postbiotic is the end product of the fermentation of the bacteria to the probiotics, which give you metabolites. So the metabolites at the end of the day are actually the postbiotic. So these are very important for our health. Let me give you one simple example, that probiotics working on prebiotics to ferment those prebiotics and make short-chain fatty acid. Your colon does not function without short-chain fatty acid. Every cell in your body uses glucose for energy, except for your colon. Your colon uses something called butyrate, which is the end product of this postbiotic term we use. That's how our colon functions. Thank you.
Okay, so today let's talk a little bit more specifically about probiotics, and one in particular, LGG. Could you tell us what that stands for and what it is?
So Lactobacillus GG is named after Golden and Gorbach, two microbiologists working in Boston, harvest this strain of the lactobacillus from a 32-year healthy young man. I love this LGG, Lactobacillus GG, or Lactobacillus rhamnosus, the scientific term. This is really a fantastic probiotics, which have so many studies done on this probiotics, including studies done in my lab.
So what makes LGG unique?
LGG is unique in the setting that it works through a protein called P40. P40, for the size of protein, is 40 kilodalton. So let me give you an example. If you take LGG and put it in a cell culture and take the supernatant and run it over gel, you will see P40 protein, which is the active component of the LGG. Why this LGG and the P40 is so important? Because there are a number of studies to suggest that P40 is given to an inflammatory state like IBD, it improves the IBD, inflammatory bowel disease. How does it work, the LGG? LGG work through P40 protein, and that P40 protein work through epidermal growth factor receptor in the GI tract and make our gut tighter membranes. Moreover, beside this, LGG also, through number of mechanisms, including increase in short-chain fatty acid, like butyrate, increases the activity of T regulatory cells, as well as number of compounds involved in recognition of antigens, such as dendritic cells. So these dendritic cells, as well as T regulators, are very vital for our immune system in the GI tract. That's what makes LGG is so unique. And the addition of LGG to infant formulas had made major advance in those formula given to our kids.
What about peptides in extensively hydrolyzed formulas?
It is clear nowadays that extensively hydrolyzed formulas are unique compounds in which they are composed of one single amino acid, two peptides, like two amino acids, tripeptides, like 3 amino acids, or 4, for example, tetrapeptides. Those are essential because it changes your gut microbiome to become a diversified microbiome. This is much better than being a restricted microbiome. So the addition of peptides extensively hydrolyzed in an infant formula make it unique in regard to changing your gut microbiome to become a healthy, diversified microbiome.
What is the impact on future allergy with these peptides and the LGG that you were speaking about?
Extensively hydrolyzed formula plus LGG present a unique formulation to prevent allergy, number one. Number two is to allow the individual who has allergy to have tolerance for cow's milk in the future. So the presence of LGG is very important in the setting of extensively hydrolyzed formula because it does a couple of things. One, it changes your gut microbiome to become a diversified microbiome. Second, it improves calprotectin, which is a marker of inflammation in the GI tract in kids who have cow's milk allergy. So it decreases the level of inflammatory cells in your GI tract. Third, if you give extensively added formula plus LGG in someone who does have cow's milk allergy, it will produce tolerance in those individuals for the future. This is an important point for you to remember this, because if you give an amino acid based formula, you are not likely to develop tolerance. However, extensively added formula plus LGG will give you that tolerance for cow's milk. How does that work? How does LGG plus extensively added formula works? It does work by increasing short chain fatty acid like butyrate in your colon, and those compounds are very important in increasing your T regulatory cells, which are good cells to have in your GI tract. So the point is here very important. Extensively added formula plus LGG is very relevant to treat patients who have cow's milk allergy, and second, it allows the patient to have tolerance for cow's milk in the future.
Very interesting. What do you wish that your peers would know about the microbiome, especially in this context of nutrition in the first 1,000 days?
Okay, I'm going to emphasize this point quite nicely by indicating to all of you that there are incredible research nowadays in the role of the microbiome, and specifically LGG, if it's given in an open window during childhood. We have evidence that in animal models, we have clear evidence that if you give LGG through its P40 protein for young animals during early life, it prevents inflammatory bowel disease in the future. The same thing for human beings. 1,000 days of a child's life, to me, is classic for the window of opportunity to give LGG during that critical time to prevent future inflammatory state in your gut. How does that work? Well, it's clear that the presence of LGG through its P40 protein tighten your membranes in your GI tract. So the epithelial integrity of your gut is markedly improved. Moreover, there is improvement in number of immunological factors in the GI tract from signals of the microbiome, and therefore there is evidence now in humans to suggest that the incidence of IBD in the future is significantly lower if you take LGG during that critical period of time. And what critical period of time is the first 1,000 days. Those first 1,000 days are so important for human health, for kids' health. Why? Because the brain increased by significant amount. Length, height, weight, everything is increased in a markedly remarkable area, and during that period of time, LGG works so well in preventing future inflammatory state. I hope I convinced you that the microbiome in health and disease is very relevant. I hope that all of you in this audience make sure that you have a good microbiome, diversified microbiome in your GI tract, so that you will not get to a problem with dysbiosis, which is a restricted microbiome, which is not good for you. I wish you the best of luck in your meeting, and I hope in the future I will meet many of you during this lecture series, which we are doing. Thank you again.
Thank you so much, Dr. Ghishan. Very informative, very interesting about the microbiome, and I appreciate you being with us today. Thank you all for joining us, and we'll see you next time.